Good morning, class. Today we study two lessons. The first lesson is about the cytoskeletons, and the second is about the cell division and cells regulations. And as I has been announced that you have another chance to improve your midterm score by present about the mitosis and meiosis. And before we begin the lessons, who wants to present today's can come to meet the midterm teaching assistant to read it for him. Tất cả các bạn nào muốn present về nguyên phân hoặc giảm phân vào phần 2 của bài này để cải thiện điểm midterm thì nên gặp anh trợ giảng đăng ký Student ID. Và đến phần 2 nếu mà số lượng vượt qua 4 hoặc 6 bạn thì cô sẽ gọi random bằng cái Student ID đó. Chứ cô không gọi tên, các bạn không phải đăng ký tên đâu ạ. Vì có tên biết đâu cô lại thường là cô thích tên này hay là không thích tên kia, nó không khách quan. Các bạn có thể đăng ký là present nguyên phân hoặc giảm phân tùy các bạn. Nhớ đăng ký rõ là present phần nào nhé, tí nữa tôi sẽ gọi. Các bạn có thể đăng ký cả hai để tăng khả năng được gọi. Lâu hay nào hai cột cho cô một tờ là nguyên phân, một tờ là giảm phân thì nó cô gọi.
Quá đông quá. Ở đâu
This is the midterm score. It will be sent to you individually next week. So I think most of you is
อ๋อ
actually from 6 to 15.
không đứa nào trên 10 á. Không ai trên 10 á. Không ai đủ 10 á.
So we started the first part of the cytoskeletons, and the second part is about the cell cycle regulations. And then in the second part, we come with your presentation, okay?
Để vậy. Tại đi em chẳng nghe được cái gì.
Keep quiet, please. We started the lessons. So in the next class, we are going to discover another part inside the cells. So our bodies have the bones, we have our own skeletons, and the cells also, they have the cytoskeleton. Cytos mean the cells. So we study about skeletons of the cells. Inside the cell, there is so many activity happens, and here's your secret turning the vesicles from the ER to the Golgis, and within the forming a small vesicle, so it transferred from ER to the Golgis, but to transfer in the right ways in the correct directions is need the help of cytoskeletons. And second, in the mitosis, I think some of you have prepared about the mitosis, and you have already studied about the mitosis in high school. So in the mitosis, the chromosome is lies in the middle of the cell, and then they push up into the opposite pole of the cells. So all of these activities is the help of cytoskeleton also. And others, this is the activity of the nervous cells. These three pictures is cut off from videos when an axons of the living neurons. So you can see that there is three vesicles marked by the red, blues, and the yellow part. And in the 6 seconds, they move. Two vesicles move toward the tip of the axons, and one is moved in the opposite pole. So how they can move, how these ties of vesicle can move in the right directions is all the help of cytoskeletons. So the skeletons have the movements of the organelles and also the chromosomes to the right place at the right times. And also, they play very important functions in the movement of the moving cells. So within our bodies, there is one type of cells can move by themselves. This is macrophages. Cells or white blood cells, they can eat the materials by changing the shape. And also the only cells can move only cell have the flagella inside our bodies is the sperm cells. And the movement of the sperm cells is need the help of cytoskeleton. Actually, the structure of the flagella of the sperm cells is the cytoskeleton also. So, what are the cytoskeletons? There is three functions of the cytoskeletons. The first, they have the movement of all the vesicles, the chromosomes, and the vesicle of the molting cell inside our bodies. And second, they have the transport, the vesicle, the compounds from this area to other destinations by the right time, the right place. And last, a very important function, structural support. So for the structural support, it is more like stimulus like our bones, our skeletons, right? So that's why we call them the cytoskeletons. And the cytoskeleton, they are composed of three major types of proteins. There are three. The first is the microtubule. You can see the big here, the big part here, microtubule. The second part is intermediate filament. And the last part is the actins. So we are going to discover very briefly about these three parts of cytoskeletons. First about the microtubule. The basic subunit that form a microtubule is we call the tubulins. Here the B1 alpha and beta tubulins bind with the GTPs guanosine triphosphate. And briefing this structure is ATP adenosine triphosphate here is guanosine, so it difference in the base here. Adenines or guanines. So alpha and beta tubulins bind with GDPs to form one subunit of tubulins. And microtubules can be cut by the cutting this one protein's name for Japanese word, and this is the scientists who discovered these proteins. Structure of microtubule. This is one subunit of tubulins, second subunit, third, fourth, and together they form a structure we call the protofilament. And here's 13 protofilament can make a holotrip. We call the microtubule. And they form this holotube with the diameters of 25 nanometer. So pay attention about these kinds of numbers because they appear so many times in the exams. Actually, the lesson about the cytoskeleton is quite deeper. So for previous year, I just revealed the information about the cytoskeleton a lot. I just emphasize the important information of this. So the functions of the microtubules are the mitotic spinner is made this mitotic spinner, so it's the big half of the mitosis. And second, it can form the core molting structure. We have a cilia and flagella. And the next it can work only with the motor proteins, and together microtubules and motor proteins can have the transport and can have the movements of the cells. And some kinds of virus, some kind of pathogens like the HIV virus can hijack this structure to bring their material to rig the nucleus, to rig the outer nucleus and replicate. So we can, by this information, we can treat some treatments of the microtubule the pathogen that's used are our microtubules and the motor proteins. And this is some treatment of microtubule of the disease that related to the microtubules. So the drugs or the chemicals that can make the microtubules less stable is leading to blood the mitosis. This is important information, and we can use them to treat the cancers. And here is one very famous we use in the laboratory cells and the positive controls is polyphase cells. Polyphase cells is natural products produced by specific plant and it can bind to microtubules and make them more stable. When they're stable is when they too rigid. It means that the cells cannot form the mitosis. So it's used to treat the ovarian and breast cancers. The second type of drugs, you can find the colchicines. The colchicines can cause the cells microtubule disappear, and we can use them to treat the gut disease. This disease caused by your inflammation. Your immune system causes an over-inflammation. So when we treat these colchicines, it can block the migration of the blood cells. So we can review the response to inflammation. And the third, we have the zosamides in the fungicides. This can bind specifically to the fungal tubulin and prevent the fungal growth. Ba cái này hơi khó, cô sẽ giải thích một tiếng Việt một tí. Cái bộ xương của tế bào thực sự nó rất quan trọng bởi vì mình có thể tắc ghép nó để chữa rất nhiều các loại bệnh khác nhau. Ví dụ như là mình có thể tắc ghép để ở đây là mình có thể ngăn chặn ung thư. Ung thư là cái sự tăng sinh quá mức của các tế bào thường mà nó tăng sinh gấp rất nhiều lần các tế bào thường của mình, đúng không ạ? Và nó là immortal cell và mình không thể nào hạn chế cái sự tăng sinh của nó. Thế thì các bạn có thấy là một cái thuộc lại mình hay người ung thư hay bị đi là gì nhỉ? Hóa trị liệu, đúng không ạ? Vâng, và hóa trị liệu là một cái chất mà rất nổi tiếng ở đây là polytaxel, bọn cô còn dùng ở trong các lab để làm đồ trứng dương ấy. Polytaxel này được chiết xuất từ cây thông đỏ, nó là natural product. Và cái nhiệm vụ của polytaxel là gì ạ? Nó bám vào cái microtubules của tế bào ung thư, và khi nó bám vào như thế thì cái gì không xảy ra? Nguyên phân không thể xảy ra, bởi vì là khi mà nguyên phân các bạn nhớ là nó phải tạo cái mitotic skin, nó sẽ kéo các nhiễm sắc thể về hai cực, mà khi cái taxol này nó bám vào đó thì nó sẽ không có cái sự nhân lên của tế bào ung thư nữa và có thể hạn chế. Thế thì bởi vì là nó tác động tế bào ung thư thì đồng thời nó sẽ tác động đến tế bào thường, cho nên là những người mà đi hóa trị liệu thì gặp rất nhiều các tác dụng phụ, đúng không? Như dụng tóc đầu tiên này, cytoskeleton của mình cũng là một cái phần cực kỳ quan trọng để tạo nên tóc và máu, đúng không? Và đau đầu này, nôn mửa các thứ. Rồi cái thuốc thứ hai là colchicine để chữa bệnh gút. Bệnh gút thì nó có liên quan đến một cái sự gọi là cái viêm mãn tính, viêm không mãn tính quá mức và nó dẫn đến sự tích tụ của acid uric. Thế thì cái việc đầu tiên đó là khi mà dùng cái colchicine thì nó sẽ block cái microtubule nó giúp cho sự di chuyển của tế bào bạch cầu, và khi tế bào bạch cầu không thể di chuyển thì nó sẽ responsive to cái inflammation này, sẽ ngăn chặn cái inflammation này, viêm cái sự viêm mãn tính này, và cái cuối cùng đó là losamize, nó chỉ là một thuốc trị nấm thôi, nó sẽ bám vào cái tubulin của cái nấm và nấm cũng sẽ không thể grow nữa, tương tự như cái taxol. The microtubulars or the cytoskeletons only work with some motor proteins. These parts I just introduced to you, and there's no appears in the final exams. Okay, so this is one type of motor protein. You can see that there's two strands, two trees of the cytoskeletons, and by the help of motor protein, these two microtubulars can move in the different directions. And here, there's so many motor proteins work in both sides. So there's two ways. When this is two sides with proteins, but they put these vesicles is move in these opposite directions. And if in which way we have more motor proteins, this vesicle will move by that direction. And the second part, both sides of the vesicle have the motor proteins, and they work by the connection, by the complex of these motor proteins. And here the microtubulars, under the microscope, all the microtubulars organized around a red point here, and they extend throughout the cytoplasm. So the microtubulars, imagine that they are a tube like this. They have two ends. One end is very dynamic, and another end can bind to microtubulars organizing centers. And we call them the MTOC. This is dynamics because it can shorter and longer in very short times. And this microtubular organizing center here in the red part, you can see under the microscope is the microtubular organizing centers. And there is two types of microtubular organizing centers. First is the centrosome. So in mitosis, there is two centrosomes in the opposite pole, and this pulls the chromosome in exposed. So the centrosome is made of two centriones, and the centrione is 9 triplicates. Nine triplicates of microtubule. So it's 9 triplicates of microtubules can make a whole tube, and this whole tube we call the centriones. 2 centriones will make the centrosome. And this is the point that all the microtubules inside the cells stick it. There are second types of the microtubular organizing centers. We have the basal bodies. So here you have a cilia flagellas, and this is the pictures of the flagella under the microscope. And the red part here, the red part is the microtubule. And here in this part, we have a core here. It's the second type of microtubule on the centers. We call the basal body. And the structures of the flagellas, who remember, is 9+2 arrangement. There is 9 double triplets. And in the centers, is it the structures you might see in the document 9+2 arrangement. And they are made of microtubule. The second important part of the microtubule or cytoskeleton, this is the actin filaments or microfilaments. So for this part, remember for me we have the protein actin. Actins will form a strings of protein actin. One string. So 2 strings of actins, they treat to form a blood helix vallis is the actin. And the function of them, maintain the cell shape, cell molting, cell divisions, and so here's the actin monomers and polymerase into the filament 8 nanometer. This is diameter 8 nanometer. The actins resemble double strings string of bed that have the right handle helical twist. So 2 strings of actins, 14 twist together to make the microfilaments. And their dynamics also like the microtubules. They can longer by the actin polymerations or shorter by actin depolymeration. And both actins and microtubules is involved in the movement of the cells, but the actin is movement more inside, and the microtubule is the movements of the molting cell like flagella and cilia. The actins is more involved in movements of the like the macrophages. And in amoeba, remember in amoeba, con trùng roi hoặc trùng biến hình is more related to the actins. And the last part is intermediate filaments. They are major components of the cytoplasmic and nuclear cytoskeletons, and they're made of various types of proteins. And again, they make 2 strings, and then these 2 strings twist together to make intermediate filaments. and they have a diameter from 8 to 12 nanometer. It's essential to maintain the core tissue structure and functions, and their structure supports resist mechanical stress. And the defective intermediate filaments cause many human disease. So they are the thus very important parts of cytoskeletons, but we still know little about them. And important function of them is just structure support, while actin is movement, and the microtubules is movement also. So the dysfunction of intermediate filament can cause many human disease. For example, here is one disease in humans related to the keratins. Thành phần quan trọng của tóc và móng luôn và da người, phần biểu mô của da. Cells are the most intermediate filaments and called the keratins, and they express in different kinds of the epithelial tissue of the bodies. So mutation in the keratins, finds or foldings is leading to the epithelial cell of flagellantes and inherited the skin glittering disorders is called the EBS. EBS, bệnh ly thượng bì bóng nước. Các bạn có thể đọc thêm về căn bệnh này ở trên Google nếu mà các bạn search ly thượng bì bóng nước. Đột biến về cái keratin, các gen keratin của mình, đặc biệt là keratin số 5 và số 14 sẽ dẫn đến căn bệnh EBS. Và ở cái thời của cô đã có gần như là một chiến dịch tình nguyện và quyên góp để chữa cho các bé bệnh nhân gặp cái bệnh này. Tức là khi mà bị bệnh này thì cái lớp biểu bì da của mình nó rất là mỏng và không chịu được áp lực hàng ngày, thậm chí là các bé mặc quần áo cũng có thể gây lên xước da và nổi những cái mụn rộp, gần như là các bạn tưởng tượng là một em bé sẽ bị bỏng gần như suốt đời ấy. Chỉ bằng một cái việc bị mặc quần áo và nó cả trà sát vào da của em bé đó. Và this disease related to the disorders of the intermediate filaments in the keratins, fibers or proteins. I will get in the second part. Our second lessons is also division and cell cycle regulation. In the first part is about the cell divisions. We have cells, we have a mitosis and also the meiosis. That is your job.
The job, man.
We study this very well studied in the high school. If you have time or you don't want to present this, I will stop you. But we now, I think there's so many volunteers waiting to present to improve their midterm score. So we will turn into the second part about the cell cycle regulation. What is the cell cycles? This is the cell cycles. We have a life cycle, and this is the cell cycle of cells. This is cell cycle of a normal cell lasts for 24 hours. And 90% is the interface yeah. Kỳ trung gian này. 95% of the cells is the interface, and what happens in the interface? There is cell grow ones. One point here is turned to DNA replication in the S phase, and then we have a cell growth 2. Here you can see the centrosomes, DNA replicates, and the centrosome replicates. By the end of the S phase, we have DNA duplicates, and the centrosome duplicates also. What is made of centrosomes? Yeah, cytoskeleton in the previous lectures. Cytoskeleton is made of centrosomes. DNA replicates, centrosome replicates, and in the cell growth 2, there are so many proteins and the cell activity happens. For this slide, please remember for me this order: G1, S, G2, and M. M is mitosis. This happened only. This is 90% of the cell. This is 10% of the time of the life cycle. And what happened in the interphase? Cell growth, DNA replication, and this is the normal activity happened in this interphase. So what controls the cell cycles? The factors that's considered when deciding whether the cell turns or moves to the cell cycle or not. We call the cell cycle regulators. And this is two factors involved in the regulate of the cell cycles. We have a cyclin-dependent kinase and the cyclins. And they only regulate on the cell cycles when the cyclin depends kinase but the cyclin. When they bind together, the cell cycle will be regulated. Here the cyclins, the scientists would discover the first cyclins when they know that the high level cyclin were associated with the onset of mitosis in embryos. Whenever the embryos perform the mitosis, they found one kind of proteins is very high levels. It is cyclins. So it is a hypothesis. Thus, this protein involved in mitosis. And based on the phase of the cell cycles, during weak-base presence, we classify the three classes of the cyclins. G1 cyclins, responsive for moving the cell cycle from G1 to the S phase. S cyclin required to init and maintain the DNA replications. And the M phase is mitotic cyclin init the mitosis. The second part, we have the cyclin-dependent kinase. Cyclin-phụ thuộc kinase. They are the method of regulator of the cell cycles. They are kinase. Whatever I written in both here is is important and it may appear in the exams. So the silicate, they are kinase. It means this enzyme that's phosphorylate or attack the phosphate group to the target protein. It is the job of these enzymes. And the cyclin-dependent kinase only work with the cyclin proteins. They are very conserved during the evolutions. So you can see that humans, there's only one silicase, but other mammals, for the deep cell, they have only one silicase, but for other mammals, they have a multiple silicase, and they use different states of the cell cycles. Here, you can see the structure. CDK1 plus the cyclin B, very important, is up here everywhere. CDK1 plus cyclin B is innate or promote the mitosis. The same way replication in the yeast cell, CDK1 plus is cyclin B. But you can see in the mammalian cells, there are so many other types of cyclins. Like innate, the replication, we have CDK2 and the cyclin E. And again, we have the graph, the same as the cyclins. And this graph illustrates that different clusters of the singers CDK activity stimulates different events during the fission of yeast cell fibers. During the mitosis, is very high level of the CDKs. So it's believed that different level the CDKs and cyclin activities requires for two transitions. Lower level is the S phase. High level is mitosis. And together, CDKs plus the cyclin, we have the maturation promoting factors. Remember for me, this structure only. The regulation of the CDK cyclin complexes, we can regulate them in several ways. First is phosphorylation, that's push or adding one phosphate group. Here is from inactive to the active form. Or second, proteolysis. You can see that when CDK1 plus cyclin B, mitosis happens. But then when your enzyme degrades the cyclin B, there is no more mitosis. Or others, inhibitor bindings. There is another type of proteins can bind to this structure and block them. Or subcellular localization. You can see that in the G2, there is no mitosis. Cyclin B will stick with the MES and bring them into the cytoplasm. But in the mitosis, it can stick with NLS, right? And bring them into the nucleus. And then the mitosis will be happening. All the other types of regulators, you need to remember this structure, anaphase promoting complex. Remember for me the abbreviation of them, APC/C. So you can see that cytochromasis will hold together by the cohensin. And in the prophase, they stick together by cohensins. In prometaphase, still stick together. And in metaphase, they need to separate, right? The separase is enzymes can provide the cohensins, but in normal state, it will stick with the securins. And this structure, APC plus C here, it can degrade securins, really be separate, and it can proteolysis the cohensin and really two chromosomes. Another thing related to the cell cycles, we have the G0 state. The cells may withdraw from the cell cycles to go to the G0 state. Or another name, we have the quiescence. Or quiescent stage. Third is we call the resting stage. Resting stage is means all the activity happens. It's normal, but just no subdivision. And here in the yeast cells, we have a start point in the yeast cell in the Z1 of the yeast cells. If they can go across the start point here, it will go to the 0 stage. And in the higher eukaryotic cells, we have the restriction point. If the cells cannot pass through these restriction points, it will go to the Z0 stage. And there is some cell types in the state forever in the Z0 stage. We have the heart cells, and very famous we have the neuron cells. They don't want to divide. Of course, if the neurons or the heart cell have some damage, they can go back into the cell cycles. But in our case, they are in the 0 state. They don't want to divide. Cells in our bodies, that's when some of the cell can last for several months. But for the neuron cells, they have the same age as you are. So when you are babies, maybe up to 3 years old, your brain is no more device, and starts from that time, your neuron cell is jump into the 0 state, and they stay there. This is because when you learn a lot of things, you need to memorize everything, right? If we renew our neural cell every day, every month, it means that you need to learn again and again and again. So it will affect our memories. But that's why for the neuron cells is one very famous examples of the cell that goes into the 0 state. When they reach the maturity, the nervous cells in 0. And there is some checkpoint of the cell cycle. This is the cell cycle. It's like when you study, if you want to move from the primary to the secondary school, there will be a test here. And you move from the secondary to the high school, it will be a test here. And you want to enroll in the universities, there will be a test here. And the cell cycle also, the cell also is, you have a test here. The cell needs to perform a test here and a test here. And all of these tests we call the checkpoint. The cell will stop and check everything. Do they ready to turn into the next stage? So at 0 stage, they check for DNA dynamics. Is or any damage of the DNAs. At the S phase, they check again the DNA damage or the replications is perfect or not. as Z02, Z2, they check for DNA damage on cells. And in mitosis, it will check if there's any chromosome is unattached to the kinetochore, and together they are ensured that each daughter cell receive a complete and intact copy of the genome. So here are the checkpoints. What happened in the checkpoint? It will find some problems. If they found DNA damage, they can try to fix it, and it cannot fix. It can prevent the cell divisions by a very famous protein we call the tumor, a famous tumor or suppressor is P53. So if they discover there's some DNA damage, this first step stop the cell cycle as Z1 stage. Second step, they will activate the DNA repair enzymes. And it not work, this means that we cannot repair, we cannot fix the DNA. It's put the third stage. It will put the cells go suicide. They will kill themselves when all the damage of DNA cannot fix. So it's how it works. It is DNA damage P53 activated. You activate the CDK inhibitor, and it will inhibit the CDK cycling complexes. The cell will stop at the G1 and then fix DNA. If cannot fix, the cell will be died. Okay. This is all about the cells or regulators. Remember for me the P53, I think it's appears so many times in the exams, and also in the next lesson is maybe we study about the cancer cells. It's very important. Or maybe we have some more minutes break times, and then we come back with the presentation students. Chắc gì em đã được gọi? Chắc gì em đã được gọi?
Chắc gì em đã được gọi hả?
Có rất nhiều bạn muốn lên và cô gọi lên được. Chắc gì bạn đã được gọi? Có tất cả các bạn ở đây lên test máy hay bao giờ?
Alo, trật tự chưa? Bây giờ sẽ là phần thuyết trình, phần nguyên phân nhé, mitosis.
Có thắc mắc là tại sao thằng này phải lên mũi điểm?
Okay, so first of all, I would like to say good morning, Doctor.
So first one, I would like to know your name, your major, and you will start after that. Okay?
So my name is Phạm Đức Lâm and I come from Biotechnology and Medicine Discovery. Before going to lecture, I would like to say good morning to every student of Gen 30 Military and Dr. Lê Đình Hương, the lecture about the cellular biology. Okay, first of all, I would like to look at about the table of content. First of all, I would like to talk about the definition. What is mitosis and meiosis? The second part I would like to describe is about the process of mitosis and meiosis. And the last one I will talk about the meaning of the mitosis and meiosis.
Từ từ, em chỉ trình bày về phần mitosis thôi nhé.
Dạ vâng ạ. Okay, so let's begin now. First of all, it's about the definition. What is the meiosis? Okay, as you can see on the slides, mitosis is the process of cell division, which help to produce two cells that have the same genetic material machinery in the cell as the original mother cell. Mitosis appear in vegetative cells and early genetic cells, especially in spermatozoa and egg-producing cells, but it do not have ability. So as you can see in this picture, I have the illustration for you about the mitosis. Mitosis is nearly to the cell division. Okay, so okay, this is about the meiosis, so I don't explain this. Okay, so next, I would like to describe to you about the process of mitosis. The process of mitosis contains 4 periods. The first period, this process is double chromosomes constituting the nuclear membrane, which occur in the cells. It occur depolarization and cell will agree. Next is about the middle periods. The double chromosome will have a maximum fluorescent concentration spread in one row in the equation, and as you can see on the box is videos about mitosis. Expecting one row in the equational plane, the cell segment stick to two sides of the chromosome at the center of dynamic. So the next period, the chromium will take place the phenomenon and of separation and movement on the cell's segment to the two poles of the cell. And last one, I think is very important. The last period, the chromosome gradually sketch the nuclear membrane appear. So at this time, the cytoplasm will divide into two total cells like you can see in this picture. Okay, so I will pass on this. Because this yes, so I will. Okay, and here is our last part of my presentation. It's about the meaning of meiosis, the meaning about the process of meiosis. Okay, so about the meaning of the meiosis for single cell eukaryotic meiosis is the reproductive mechanism. Okay, as you can see in this picture, this illustrates for you about the meiosis. Okay, another thing is about for the multicellular eukaryotic, the process will increase the number of cells and helps them to grow. Furthermore, it helps the body to generate the damage of the tissue of the cell. And that is the end of my presentation. Thank you, Dr. Lê Thương and every student of Gen 30 Marie Curie. So, do you have any question for me?
Ai muốn comment không? Does anyone have a comment or some question to
Cái
ask?
bạn nào xanh ngồi ở dưới góc dưới kia có phải có giơ tay không ạ?
Em hỏi tay đi. Hiện tại cứ đi đấy mà đi bắt tịch đấy.
Cho mình hỏi là bạn áo xanh muốn hỏi trước hả?
Vâng, hai thì vị.
I have a question. So you explained about the mitosis, right?
Yes.
So in your opinion, how can mitosis make cells that have more than one nucleus?
Can you say again your question and please talk slowly?
Okay.
Slowly.
How mitosis can make cells that have more than one nucleus?
So your question, okay, I understand your question. That's a little complicated
Ta
for
không hiểu
me.
về câu hỏi sinh nghe.
You say that with one nucleus, so how can the mitosis can make another cell? That's right. That's your question.
I guess.
Okay. So I think during the process of cell division, the nucleus, I think the nucleus will divide into two.
No, it's okay. We can elaborate on that later, I guess. I would like to comment on your presentation.
Okay.
First of all, I think that you may have used too many templates on one presentation, so the overlook looks very confusing and it doesn't gives the feeling of a whole presentation. It's like a work of many people work on one PowerPoint, and so it looks kind of messy to me. Second of all, I think that your presentation might have too much word on the on slide, I guess, because I cannot focus as on what is the most important or the main ideas or things that you're explaining. So you're just putting words on that. I can't see anything. I don't even understand like the illustration you're giving because I don't know what does it illustrate for.
Okay.
And I think that generally you may have many và tiếng Việt nhé. Cậu nói tiếng Anh một số từ hình như là cậu nói sai pronunciation. Hold on.
Thank you.
Hold on. I noticed that you're pressed on time, but I think that you should elaborate more or emphasis more on the part that you need to focus on, such as the process, and also I think that you may have summarized too much. Wait, no. So the information that you're giving us is maybe it's not enough to me because it looks like it's just like definition you're finding on the internet, and I don't see any like specific things that you're pointing out. It's all the general thing, and I expect the university to give us more detailed things in high school stuff. And well, overall, I think that the presentation is great. You have the courage and also the tinh thần làm việc cố gắng. But I wouldn't say this is the greatest thing I get. Yes. Thank you.
Wait. Okay, so first of all, I would like to say thank you to your comment. Nói về trước này. Cái này thì mình thừa nhận là thứ nhất mình có sai pronunciation. Cái này thì mình sẽ cố gắng mình về để mình nghĩ. Cái thứ hai đó là bạn nghe có nói là cái bài của mình có nhiều chữ, mình lấy ví dụ như là slide số 4 này đi. Cái này thì mình cũng đã cố gắng thu gọn hết sức rồi. Thì mình cũng thừa nhận là nó cũng có hơi dài thật và cũng không có cái gì ạ? Main cái chính á. Thank you very much for your comment and question. Okay. Anh thấy cái mic cho các bạn nào bạn nữa.
Okay, so I want to have another comment on your presentation. Well, can you take to the part that you are talking about the phases of mitosis? The first period, second period, or something that you write it.
Okay, so what do you want me to change my presentation to what part?
Những cái phần mà ký giữa của cậu ấy.
Ký giữa của cái mitosis hả?
Mấy cái phases của cái đấy.
Đây đúng không?
Yeah, I would like to have like an opinion that in mitosis, as a research, we don't call it first period, middle period, we call it, or something like that. We call it prophase, metaphase, or anaphase. That is, I think that if you just Google it, and you can easily find the name instead of just putting it translated directly from Vietnamese to English, and that is not how we call it in academic way. Yeah, and that's also.
Okay, so your comments about cái biểu diễn của mình đúng không? Đó là mình nói kiểu first rồi middle này nó không được kiểu khoa học đúng không?
No, you are talking about the phases of mitosis, right?
Yeah.
And you have like you want to pronounce, want to see list all the phases, but you see that they have a name, and you can, and like I know that you want to have like to separate it into different stages, but I think that you should include the name of each species instead of just pointing out your opinion like that. Yeah.
Okay, I think I forgot about that. So thank you for your comment.
Oh, and I also have a question.
Okay, so go on.
I don't think you have included some certain phrases such as the introvert phrase or something like that. So you always say this, the cells that you're talking about, it's just about eukaryotic cells, right? Cells that do not have a cell wall, but in reality we have like fungus that have a cheating cell wall and plants that have a cellular cell wall. So how do you think that they could multiply with the cell walls remaining intact after mitosis?
Can you say again your question?
Oh no.
I can't hear clearly.
Okay, so how do you think that fungus cells and plant cells can do mitosis and with their cell walls? Do they destroy the cell and mitosis or they just multiply with the cell walls on?
Okay, so your question is
Ôi,
about
khó.
how the cells is mitosis.
In fungi.
Kiểu đây là một cái chuyện nhá.
One of the videos.
Về tế bào thực vật và tế bào nấm ấy, cô đã học là nó đều thành tế bào đúng không?
Đúng, có.
Thì đây là tôi đang nói về nguyên phân ở một tế bào nhưng mà nó đã không có thành. Nhưng trong thực tế thì hai tế bào đấy nó đều có thành, và có nghĩa là để nguyên phân tế bào đấy thì cái thành thì nó sẽ phải làm như nào để sau khi mà nguyên phân tế bào đấy chia được hai và thành vẫn giữ nguyên.
So your question, I understand, but I want to say that is, thì câu hỏi là nếu mà có cái thành đấy thì để nguyên phân như nào đúng không? Thì theo tớ nhớ ở trong lecture thì để mà nguyên phân được thì nó sẽ phải có cái đường để nó đi vào. Đấy. Thì khi mà hai cái tế bào nó gặp nhau này, là một nguyên phân đúng không? Thì nó sẽ đi qua cái kênh protein.
You have misunderstood the mitosis. Mitosis is one cell became two cells, not two cells became one cell.
Okay. So I think your question, I will come home and find more information about it.
Yeah, đúng rồi.
Okay.
Đây, chỉ có thứ rồi.
So anyone have a question or comment?
It's a big body that how we know there are there any indicator or mechanism to recognize how many mitotic divisions that the cell has undergone?
The question is how many mechanisms does the cell can mitosis?
Are there any mechanism or the way to recognize how many divisions that the cell has undergone?
I think just only one mechanism that the cell can exist.
How many times that the cell has
I think it's just one time. That's how I mentioned in my presentation.
how many times when you love it?
Ý nó hỏi bao nhiêu lần là nó nguyên phân đúng không? Ok. I think the cells just only let me think.
cò này khó mày đi hỏi tới vào có thể nguyên phân nguyên lần.
I think it's so many times. So many times. Okay, how about my disease?
Thì thật sự thì nó thuộc vào cái
I have a comment for your presentation that instead of trying to summarize we can alternate it into a graph that we can learn it like easier and it's shorter.
Okay, thank you for your comment. I would like to ask, I'm really sorry because I cannot reply your question clearly.
Ôi, cái này tao
Có
hỏi
ai câu hỏi không?
bánh chiên nó có cái vẫn còn kìa cái tem á.
I think the slide is very lively and the presentation is very good and excellent. Okay, I have some comment. Thank you.
Thank you.
Để bia này.
I think I agree with the I don't know the names, the two parents. You have a very
Phải là trigger.
lovely recommends. And so first of all, your presentation is you use too much templates, and sometimes not like the work of the scientists, so many things. And the most important things, too much words, and I think you like academic words in your side, in your presentations. Like in mitosis, actually you need to remember for me key maths, P for prophase, M for metaphase, A for anaphase, and T for telophase. But these do not appear in your presentations. It also you need to mark your slides by the numbers, so when the audience wants to ask you about a slide, they can say, okay, come back to slide number 8 for numbers. It's easier for the audience. And another comment, I think you put some videos in your slide, right? But it do not work. Whenever when we talk about the anaphase, what you said the first phase, but there are no animation, so we talk about okay, the nuclear membrane disappears, nothing happens in the right passage or slides. And about the question, I think you some question you don't understand your friend's question, and no answers work. No answers were working by groups, and I just have one question. Do you want to replace your midterm test with the results of the presentation today?
So I think with the red line, the question, I cannot do it. It may get I receive a low point. So you take the test on the model as my midterm test.
No, you don't have the right to check against. You need to decide whether you take the results of the presentation today or the results of the midterm test.
I would like to take the result of my midterm test.
Okay, thank you very much.
And once again, I would like to say sorry to the person who asked me a question that I cannot answer.
So let's say sorry.
So thank you, Dr. Lenico, and every listen to my presentation.
Bị các bạn dập quá không dám lấy điểm.
Bị đúng mấy đứa không được ưa lắm, không ưa thằng này.
Biết được bao đứa.
Đúng mấy đứa combat hôn
Cuối mùa diễn viên rồi mà không được.
Đấy. Ai? Ai đây?
Một ba một rồi.
Đăng ký xong sủi. Đăng ký xong sủi. Đăng ký xong sủi. 2B1
Ai bạn nào có số này?
132. Sợ không dám lên. Sợ rồi. Rén rồi.
Mẹ các bạn chả giữ khách. Chả giữ khách gì cả. Ôi, nhiều cân.
160 có ai dám không? À kia có thanh niên.
Các bạn lại chết thề.
À không, đây thì
Who has this ID number?
Cảm ơn em.
không dám. Đủ quá không dám.
Bạn lại bị căng thẳng.
À đâu có rồi. Một thằng cùng lớp.
Rồi. Tuyệt vời.
Tôi nói trước là đừng vi phạm nhiều. Cùng lớp tôi học tiếng Anh
Ít ra nó có tinh thần lên.
có tinh thần nhưng mà
10 điểm.
phát âm không tốt đâu.
Xúc cảm.
Nhưng mà tôi nói trước là phát âm không tốt nên là chịu khó, chuẩn bị tinh thần.
Bạn nhẹ tay thử.
Nhưng mà phát âm nó có vấn đề thật đi, nó phát âm như kiểu thằng này đã học tiếng Pháp rồi.
Nhiều kia nó không được pháp.
Không, nhưng mà nó cho học tiếng Pháp rồi. Nó không biết.
Đọc tên bố.
Mạnh khỏe nhé bạn. Ôi trời. Quay gacha. Ôi trời. Mấy giờ rồi? Hôm nay sẽ tan sớm.
Các bạn
Chắc là tan sớm. Ở bên cái mic lên hồ. Đâu?
Và rất chúng tôi là you should present the mitosis part, okay?
Mitosis, am I right?
Vừa đây có bạn nào đăng chỉ đăng ký mitosis thôi nhỉ?
Nghe tiếng. Có nghĩa là sẽ có nhiều người thích trình cùng một cái.
chả giữ khách. Mới thằng này biết.
Do cái máy cắm bị kêu tiếng rẻ rẻ.
Bạn sợ quá.
Tuyệt vời, học gì cả. À không học. Rồi. Này quay à?
Who is BA12020?
À, lại xoay à? Sao lại xoay rồi? 552 lên hướng sang các em này. Quay em nữa. Boss nhiều phết. Thằng này là gì?
Em có chuẩn bị
Tôi nghĩ là bây giờ chuẩn bị cái này nào. Có tinh thần nhưng mà không được phát biểu.
Cái máy nó sợ. Cái máy nó cảm nhận được nguy hiểm.
Không có cơ hội được nói luôn. Buồn đấy. Chắc là tao cứ chơi quả slide trắng rồi vậy. Không Canva, không biết nữa. Chỉ năm 2.
So please introduce yourself and you can start the presentation.
Okay. Hello everyone, my name is Mai Sui and I come from Vietnam. So today I want to talk about the mitosis progress. Before start my present, I have some question. So we know we are made of the cell. So how starts from a little tiny cell. So how does that cells become a baby, and how does that baby become a human? To find the answer, next slide. My presentation has three parts. The first one is the cell cycle, the second one is about mitosis, and the final is the functions of mitosis. Before understanding what is mitosis, we have to know what is cell cycle. So cell cycle is a series of events that take place in a cell as its grow and divide.
Nói chung là mình đi nói đi.
So a cell cycle has a different phase called G1, S, D2, and M. A cell spends more time in interphase. At this time, the cells go and prepare for the cell division. And in F phase, the cells copy all the DNA in the nucleus. After that, a cell has have two nucleus and two centrosomes to become a M phase Okay, N phase stands for mitosis. Mitosis is divided into four main phases: is prophase, metaphase, anaphase, and telophase. Then it's changed to the process called the cytokinesis. Now let's move to the part 2 is mitosis. Other mitosis begin in the prophase. The chromatins contain to form a classical chromosome structure having two sister chromatids linked by a centromere. At the same time, the protein chain called microtubule appear. Here and the centrosomes move to the opposite pole. And the nucleus is disappear, and then the nucleus membrane is also broken down, and the centrosomes go to the opposite pole, and the microtubule go to attack the centromere of its chromatid. The structure of the microtubule attack is the protein structures called kinetochore. So next, in the metaphase, the sister chromatids lies along the center of the cell and forming the unique metaphase plate. Okay, it comes to anaphase. The sister chromatids separated and separated by the shortening of microtubule and the move the part of the pole. This one, okay. And the final phase in the mitosis is a telophase. In this phase, the microtubule is broken up. And at the end of telophase, the chromatid is moved to the ends of two poles like this, and also sorry. Yeah. And the membrane, and it comes to the process called the cytokinesis, and the membrane, and also the membrane and the nucleus appears again. That's the cytoplasm divides until we have the two distinct cells, two symmetrical and distinct cells. In summary, I have some figures to you to easy to remember this process. In the interphase, the DNA is duplicated, and then in prophase we have two main events. The chromatins condemned to form the chromosome structure, and the nuclear membrane is disappeared. And then it comes to metaphase. The chromatins align at the center of the cell to make a metaphase plate. And then on the interface, the sister chromatin is separated and goes through the opposite pole. In the end of mitosis is telophase. The event of this phase is opposite to the prophase. You can see in the prophase the chromatins contain, and in the telophase the chromosomes decontain to form the chromatins. And the nucleus and nuclear membranes reappear again. Finally, we have two genetically identical daughter cells. Each of them have the same as its parent cell. The next part is some functions of mitosis. In the first questions, I asked about how the cells become a baby and how the baby can become an adult. So this is answer the functions of mitosis is have in the development of an organism, and in a single cell organism is a process asexual regulation. Okay, and also it have to replacement from a damaged tissue when you have hurt, and the cell can replacement to the new cell to What do you experience of my presentation? Thanks for listening. Thank you. Do you have any questions or comments from me?
Hello. So, your presentation was good, was excellent, and I feel every connection between each of the slides. And you don't put many words on the slide, which is actually good since the audience will listen to your speech. But I hope that if possible, next time if you go on the presentation, you can be more confident since your voice is kind of small. But overall, I think the presentation was good. That was my opinion.
Yes, thank you.
Okay, so I have a question for you, but I have some of your comments. First of all, I think your presentation was very good. It's very simple, but it doesn't have the main beginning of the means. Your presentation good, but I have some mình có một chút góp ý là first of all, it's your body language. I see you feel shy. Yeah. And I want you to improve your shy. Sometimes I hear you breathe. Just take off your mask so it can breathe. Thank you.
Does anyone else have a comment?
Any question?
Any question? Any comment?
Không phải tự nhiên bị bắt lỗi.
Alright, thank you very much for your presentations. I think it's much better than the previous students, but it needs to improve, especially I think you need to practice more. And about the scientific more system with I think is mainstream for you, so the pronunciation is not correct, like kinetophore. Please back to the slide. So, pay attention class, when you perform a presentation, please note the number of the slide. So when the audience want to ask a question, they can even easier to find back in the slide. Back to the slide, I think about the anaphase, please. This one. So, notice schedule. The previous one. This previous one. I'll stop here. The thing that you know, the microtubules, what is this? Yes. The microtubules, you know that the microtubules, what it is? The yellow ones, the blue ones. No, the blue. Yes, the blue one is microtubules, and stop there. Okay. Okay, so what is the structures like the suns?
Now the cell circle is the centrosome, and the microtubule is appear from centrosome, and because it's the beginning, so it's the leaves is short, and
I think you need to note that in the centrosome, because microtubule is a very small part of its centrosomes. It's more detailed about centrosome. It's made of 2 centrioles, and the blue part is the mitotic spindle. If you highlight these microtubules, I just understand that all the structures, it's like the sun here, is microtubule, is not true, right? The chances about how you notice the pictures.
yes.
Next slide. Always. Right. So, what is the kinetophore? What is kinetophore?
The blue circle, the small blue circle in the So the skin transport with the proteins to microtubules attack with the chromatin. So
this center is more precise if you say that it's attached to the centromere of the chromosomes.
the centromere is a part.
Yes, but the kinare cord is appearing in the centromeres. It attached to the centromeres. And for last, we have some magnetic spinors attached to the kinare cords and pull this chromosome separate into two parts and move in the opposite poles of the mitosis. Next. Metaphase, please. Please back to the previous slide. There you go. Metaphase. So in humans, how many chromosomes can you count in one dividing cell in the metaphase?
I'm sorry, can you repeat again?
For example, if it's human cells and there is in the metaphase of dividing cells, how many chromosomes can you count for? So normal cell, in human normal cell we have 46, right?
Yeah.
So in metaphase, how many chromosomes?
Thì trong thế mà đang được bao nhiêu cái ribosome.
Uh, 92.
90?
Chromosome 92.
92? No.
46.
It's still 46 chromosomes in the 92 chromatids. So okay, maybe we don't have a lot of time. Thank you very much for your presentations. I think it's much better. You understand what happened in mitosis, or you understand the structures, endosome, kinare cord, everything, but I think you need to practice more. You're too shy to present some things. So thank you very much. Maybe it's one more student about the mitosis, and then we turn into the meiosis. So, trở lên hỏi đi.
Ai cũng đơn hết rồi. Được, kiểu đơn.
Bạn nào muốn lên mitosis thì nhờ hỏi vậy đi. Lắm thằng của chuồn mất rồi. Đăng ký rất là máu xong đi mất. Ví dụ đầu tiên không gì phải sợ cả, vì bạn nào chắc gì bạn nào hơn mình đúng không?
Lên là mình đẩy được cả cái chị kia. Mà có không có chết rồi đó. Tiêu chí chấm của là gamma state. Gamma.
Hello everyone. Today I will talk about mitosis. Maybe mitosis. My name is Long Wu. First one is mitosis. Mitosis is just one part of the cell cycle, the life of a cell from the time it is first formed during division of a parent cell until its own division into two daughter cells. Mitosis is conventionally broken down into 5 steps: prophase, prometaphase, metaphase, anaphase, and telophase. First one, I talk about the prophase. In prophase, chromatin become more tightly coiled, condensing into discrete chromosomes, and the nucleoli disappear. You can see no nuclear in here. The duplicated chromosome appear as two identical sister chromatin. The mitotic spindle begin to form, and the centrosome move away from each other by the lengthening microtubule. And secondly, I talk about prometaphase, the nuclear envelope fragment. The microtubule from centrosome can invade the nuclear area. Chromosome become more condensed. Some microtubules attach to kinetochore, which jet the chromosome back and forth. Next is the metaphase. Centrosome are opposite poles of the cell. The chromosome arrive at the metaphase plate. The kinetochores are attached to kinetochore microtubules coming from opposite poles. Next is anaphase. It's the shortest state of mitosis. The coenzyme protein are clipped, which allow the two sister chromatin become an independent chromosome. Two new daughter chromosomes more toward opposite end of the cell, and their kinetochore microtubules shorten. By the end of anaphase, the two ends of cell have identical and complete correction of chromosome. And next is telophase. Two daughter nuclei form in the cell nuclear envelope. Arise, the nucleoli reappear. The chromosome become less condensed, and mitosis, the division of one nucleus into two genetically identical nuclei, is now completed. Next is cytokinesis. Two daughter cells appear shortly after the end of mitosis. In animal cell, cytokinesis involves the formation of a cleavage furrow which pinch the cell into. Here is the process of mitosis. Next, I will talk about meiosis. Meiosis is a type of cell division in sexually reproducing organisms that reduce the number of chromosomes in gametes. Several steps of meiosis closely resemble corresponding steps in mitosis. There are two states such as meiosis 1 and meiosis 2, more than different from mitosis. These two divisions result in 4 daughter cells, more than 2 daughters in a cell of mitosis. First, I talk about meiosis 1. Phase 1, the centrosome movement, spindle formation, and nuclear envelope breakdown. Each chromosome pair with its homolog, elite gen by gene, and crossing over occur. And in metaphase, pair of homologous chromosomes are arranged at the metaphase plate, and one chromosome of each pair facing each pole. Each pair has lined up independently of other pair. Both chromatids of one homolog are attached to kinetochore microtubules from each pole. Here is the picture for prophase 1 and metaphase 1. And phase 1, the breakdown of protein allows homologs to separate, and homologs are guided by a spindle apparatus. Each chromosome moves as a unit toward the same pole. Telophase 1 and cytokinesis come next. Each half of a cell has complete haploid set of duplicated chromosomes. Each chromosome includes two sister chromatids. One of both chromatids includes region of non-sister chromatid DNA. Cytokinesis occurs with telophase 1, forming two haploid daughter cells. In animal cell, the cleavage furrow form, but in a plant cell plate form. Almost all chromosome duplication occur between meiosis 1 and 2, but in some species, chromosome duplicate occur between meiosis 1 and 2. Next is meiosis 2. The professor, almost all people think that meiosis 2 is basically similar to mitosis, so I will skip and talk about the difference. Is the telophase 2 and cytokinesis. In telophase 2 and cytokinesis, the nuclear form chromosome decondense and cytokinesis occur. Parent cell produce 4 daughter cells, each with a haploid set of unduplicated chromosomes. The 4 daughter cells are genetically distinct from one another and from parent cell. Here is a picture for anaphase 2 and telophase 2 and cytokinesis. And here is the process of meiosis. Thank you for listening, and if you have a question, contact with this email and I answer in detail.
Any question or comment?
Chim lợn hỏi ít chim lợn. Chúng ta có hỏi chiến thuật.
So, before asking you a question, I will,
Have a comment?
if you want, I would like to have some comment about your presentation. So, compared to the first presentation, yours even more worse than I expected, and the explanation was too long. And there are some person who listen to the presentation, they cannot follow your present. So, if possible, next time if you come and present your work, may have less word in the slide. And so for the question, I want to ask that how can sister chromosomes stay together and separate normally in mitosis?
The answer is in slide. If you want, I can send you.
Nội chiến, bắt đầu nội chiến. Bắt đầu nội chiến.
Can you show the slide for the answer?
No, because if she want, I will give she in the end of class.
Cứ show đi xem nào. Thầy trợ giảng bảo show thì show đi. Cô đấm đây. Cô nói mày
It's not
show.
the answers. Để hỗ trợ thế thì câu hỏi câu nha. What is the haploid?
The haploid is đơn bộ.
What is the
Đơn bộ tức là nó bằng một nửa cái lưỡng bộ. Người có bốn bộ cái thể là 16 thì đơn bộ đó là 23. Đơn bộ đấy ạ. Đơn bộ đấy.
Nó hiểu quá. Nó bắt đầu đi. Nó hồi mình lại cái. Nó hồi mình lại cái.
So in the anaphase ones of the meiosis,
Có thể thử
how many chromosomes can you count
Nhưng mà hơi mong
for the human cell?
Đấy, tao gặp mấy đứa chân đầu đấm mình.
Assuming that you have one's normal human cells in dividing stage, in anaphase ones, how many chromosomes can you count?
In anaphase 1, the parent cell is not separate into two cells. So in human, there are 46.
46 chromosomes?
Yes. 46.
You sure?
No.
92 chromosomes.
92 chromosomes. So how many chromatids?
46. Chromatid is 184.
Đúng rồi.
How do you count one subject is chromosomes and one subject is chromatids?
Chromosome is chromosome. I don't know how to explain.
So what is the structure decomposed here?
Here is this the chromatid?
No. Cái phân màu vàng này, hai cái màu vàng, hai cực là gì?
Cái đấy là trung tử, nhưng mà tiếng Anh của nó hình như là cái gì ấy em đây nó là cái cytosol.
Sensor. Rồi, được làm bằng cái gì?
Vi ống. Microtubule.
Tập vừa dạy xong rồi trong giờ những ngồi xem đây rồi.
Em có nghe mà em nốt lại rồi.
Yeah, microtubular. Xem nào, kiss the phone.
Học sinh này sẽ là đứa bị cô đấm đầu tiên. Em ăn tí nhá.
So what made the diversity of organisms in this planet?
Cái mà
Cô nhận xét luôn nhá, bài của bạn như bạn thì đã nhận xét bạn viết quá lắm chữ, đáng lẽ cái phần chữ này song song bạn để hình vào đó, quá lắm chữ. Và cái nữa là thôi thực ra thì cái phần present của bạn cũng không quá là đánh giá nhiều đâu, nhưng cái phần trả lời của bạn nên chấn chỉnh lại cái cách trả lời của mình. Cô nói thật, nó thể hiện sự không tôn trọng cô và không tôn trọng các bạn. Nếu mà trong slide có câu trả lời đấy, bạn có thể trả lời ở đây trong phần này, bạn có thể nhìn này, chứ không có cái gì mà ở trong slide để tôi gửi cho bạn chả bao giờ đi. Bạn đang trả lời thì bạn kiếm điểm chứ không, bởi vì là cái điểm thì nó sẽ cấu thành từ cái performance của bạn, là có cách bạn chuẩn bị slide, cách bạn trình diễn và cách bạn trả lời cơ, hiểu không? Còn cô hỏi vài câu thì có thể bạn cũng nhớ một vài phần ở tiếng Việt ở high school, nhưng mà bạn không biết diễn đạt bằng tiếng Anh, không biết chung tử là gì cũng được, biết crossing over tạo nên diversity cũng được, nhưng chromosome chromatic học lại nhân lên là 190 mấy luôn. Rồi về chỗ một câu hỏi, bạn có muốn thay thế điểm này bằng điểm?
No.
Trong lịch sử cô chưa bao giờ cho bạn nào present dưới 10 điểm đâu.
Đứa làm tạo nên lịch sử ấy. Ai tạo nên lịch sử dưới 10 điểm?
Cái bạn nam muốn vậy đấy nhỉ? Em muốn lên nữa không? Cái bạn nam vừa đấy lên rồi xuống ấy.
Đâu rồi?
Thế cả.
Tuổi à?
Có lên nữa không?
À, ok. Chưa. Thời đi cay. Bị hỏi loạt câu cay. Sau đó hỏi chẳng hạn. Ra ngồi chơi tí nhé. Mình có thể đặt cái 100 đeo. Đây. Click to add title. Còn nào.
Hello everyone, my name is Minh. Now I will introduce about mitosis and mitosis. So the first is mitosis. The concept is mitosis is the process of mitosis, the process of create two cells with the same genetic machinery in the cell as the original parent cells. We have about 6 mitosis periods. First is intermediate period, and next is the beginning of the first term, and this is the end of the first term, and is midterm, and is the next midterm, and is the final period. So I will talk in four periods. The characteristics of the first period: chromosomes shorten, the nuclear membrane swell and beginning to disappear, colonies begin to form. And this picture to see. And second is midterm features. Chromosomes are maximally shortened and line up in a row. The colonies space is fully formed. Nuclear membrane disappears to release chromosome. After midterm is next period. Each double chromosome split into 2 chromosomes and move to the outside end of the cell. And the last is the final term. The colonies disappears. The nuclear membrane appears to enclose the chromosome, and the last is bring isthmus cell from two daughter cells. And I want you remember the important thing is we have parent cells, and in mitosis we can have the two daughter cell, and it have the same chromosome is 2N. That's so. Next is meiosis. Meiosis is a process of cell division that occurs only in mature sex cells. About 2 is sperm and egg product cell, and resulting in the production of gamete, sperm and egg that carry half of the cell's chromosome set of mother give evenly. And these two types of mitotic, we can see this is a chain in 4 periods, and the result of meiosis from parent cell through mitotic, we have 4 daughter cells. This we have a parent cell, but in meiosis we have not have 2, we have 4 daughter cells. But it doesn't have the same set of chromosomes like parent cell. The same set of chromosomes like parent cell just have haploid. Haploid is như bạn Long Phú vừa nói, đó là đơn bội. Xin lỗi nói tiếng Việt, em xin phép cô nói tiếng Việt. Cái này là đơn bội là chỉ có 2 n, thì là 4 cái này chỉ có n. Đơn bội chỉ có n, 2 n. Có khác với cả cái phần mitosis, cái phần phần nguyên phân đó là ở đây thì nó sẽ là 2 n. We have different between mitosis and meiosis. Alike all have mitotic spindles. The second division of meiosis takes place like mitosis, and the difference is mitosis occur in vegetative in primitive sex cells, but meiosis occurs in sex cells during maturation. And the last mitosis include 1 cell division, but in meiosis consists of 2 consecutive cells divisions. And important difference you can see in mitosis as 2N, and meiosis we just have haploid. Thank you for listening, watching my presentation. Do you have any question for my presentation? I'm from mechatronic engineering technology, so my English in biology is not perfect. So I hope you can ask me Vietnamese.
Anh muốn comment không ạ? Anh muốn hỏi đúng không?
thì trong quá trình nguyên phân với giảm phân ấy thì nó sẽ có những cái lỗi xảy ra, và không biết là những cái lỗi này thì nó sẽ kiểu gây ảnh hưởng gì đến cái gen của mình, và nó có thể tạo ra những cái loại bệnh gì, thì bạn có thể đưa ra một ví dụ cụ thể không?
Nghĩ là nếu mà xảy ra vấn đề hay lỗi lầm gì thì có thể nó sẽ bị thay đổi gen, như kiểu là nó không còn giữ nguyên tình trạng gen như ban đầu nữa. Tôi nghĩ thế.
Cụ thể là có thể gây ra căn bệnh gì?
Căn bệnh thì nói bệnh thì có nhiều bệnh, mà cụ thể ở đây thì tôi nghĩ cái bệnh nguy hiểm nhất nên là nó sẽ mất cái điểm sắc thể thông tin ban đầu. Xin lỗi em, nhưng mà kiểu mình không ở trong chuyên ngành sinh nên là cái knowledge của mình nó không được rộng lắm. Và mình vẫn cố gắng trả lời hết sức có thể những gì mình biết từ trong qua 12 năm học vừa rồi.
Anh có câu hỏi cho em là ở giảm phân từ một tế bào giảm phân ra thành 4 tế bào đúng không? Vậy thì đúng không?
Đúng ạ.
Vậy thì một tế bào sinh tinh sẽ ra bao nhiêu tinh trùng và một tế bào sinh trứng sẽ ra bao nhiêu quả trứng?
Ra bao nhiêu tinh trùng ạ?
Một tế bào sinh tinh thì sẽ sinh ra được bao nhiêu tinh trùng và giảm phân được bao nhiêu thành được bao nhiêu tinh trùng, và một tế bào sinh trứng giảm phân thành bao nhiêu quả trứng?
Em nghĩ là tất cả đều là 4.
Nếu một lần một quả
4 trứng 1 tinh trùng.
mỗi lần giảm phân mà ra được 4 quả trứng thì thực ra con người toàn sinh 4 để sinh
Con nhầm 4 tinh trùng 1 trứng.
Vâng, cảm ơn bạn rất là cố gắng. Bạn có muốn lấy điểm này theo như điểm biết không?
Chứ nếu mà sang điểm 20 thì em được bao nhiêu điểm?
Bạn không được biết trước điểm. Tự cảm nhận thôi.
Thực ra thì cái hôm mà kiểm tra midterm ở kia thì em thật sự là bài của em có thể dùng vừa nát bét. Nên là đấy cô bảo là cô chưa cho ai điểm thi danh dưới 10 nên là thôi em lấy.
Bạn ơi,
Okay, so the lesson today is finished, so you can come home right now. Thank you.